Hello friend, I am Dr. Sirajul Akfazi. I welcome you in my YouTube channel. Today in this video, I am going to talk about mechanism of enzyme action. So मेरे दोस्तों enzyme actions की mechanisms क्या है? How enzyme act? So according to Michaelis-Menten hypothesis, enzyme molecule आप उसको E मान लें, फर्स्ट कंबाइन विद सबस्ट्रेट मोलिक्यूल उसको आप S मान लें. तो E is the enzyme molecules and S for substrate molecule. To form an enzyme substrate complex उसको आप ES मान लें, which further dissociate जो बाद में टूटता है to form product उसको P मान लें और enzyme यहाँ से निकल जाता है, so उसको E back मान लें. So my friend, enzymes are biochemical catalysts involved in the chemical transformation of a specific compound or a group of compounds, the substrate. It has been established enzyme activities depend upon the presence of various metals. The metal in an enzyme is present as a chelate on the basis of the easy with which the metal can be removed from the enzyme. So Valley एक साइंटिस्ट का नाम है, Valley designed them as metalloenzyme inert complexes in which the metal does not leave the enzyme easily. इसमें जैसे मेटल और enzyme बना तो उसको metalloenzyme कहें. Even in the presence of large amount of good coordinating agent and metal enzyme complexes, labile complexes बनते हैं, from which the metal can be removed easily. तो आप लेबाइल का मतलब समझ जाएं, जिसमें मेटल इजीली निकल जाए उसको मेटल लेबाइल कॉम्प्लेक्स कहेंगे, और जिसमें नहीं निकलता उसको इनर्ट कॉम्प्लेक्स कहेंगे. The former involve a specific metal and analogous complexes with different central metal ion having similar charge and size, which do not behave catalytically in the same manner. In the metal enzyme complexes, the metal requirement is usually of a more general nature and may be satisfied by a range of ions having similar charge and size. तो ये बहुत इम्पॉर्टेंट है कि सिमिलर एटम टाइप का मतलब है इन केमिस्ट्री आप उसकी चार्ज और साइज. चार्ज क्यों? एक ही उस पर चार्ज लगाने पर उसकी साइज को आप कांस्टेंट कर सकते हैं. Thus, magnesium 2 and manganese 2 are often both effective as activator of many enzymes. The metal ion which are found in various enzyme systems are iron, copper, manganese, zinc, magnesium, molybdenum, and calcium. My friend, the another important topic is transition state theory. Transition state theory. The word saying transition state theory. The formation of enzyme substrate intermediate. Enzyme substrate intermediate means formation of ES. E for enzyme, S for substrate. Complex during enzyme activity in physiological system is very complex, and the precise mechanism of this interaction is almost unknown. It is reported that the activity is a combination of steric, electronics, and pharmacokinetics factors, and it could be understood in the light of chelating theory. Participation of a specific metal ion during intermediate complex formation, irrespective of whether a metal ion forms the integral part of the active site of an enzyme or the enzyme requires the metal ion as a cofactor for its activity. My friend, mixed ligand complex involving metal ion, enzymes, and substrate are formed in the most enzymatic reaction. The specific role played by a metal ion in an enzymic reaction is often difficult to elucidate due to the complex nature of the protein structure. The model studies with relatively simple molecules of known structure often yield valuable information that gives clue to the roles of metal ion in many enzymatic reactions. So metals are supposed to serve two purpose: provide proper stereochemical orientation first, and the second, bring reacting molecule, the enzyme and substrate, closer so that the reaction may occur. So, my friend, three possible probable mechanisms have been proposed to explain the mode of action of enzyme. Metal ion forms complex with the protein part of the enzyme apoenzyme, forming the active enzyme EM. The active enzyme react with the substrate to give intermediate. So E plus M it give EM. Enzyme metal substrate complex EMS, which subsequently decomposes into product P derived from the substrate S, an active enzyme. For example, EM plus S is give EMS. EMS undergo dissociation, it give EM plus product. So, the second case, the metal ion forms complex with the substrate first. The metal substrate complex MS react with the enzyme to give the enzyme metal substrate complex EMS, which subsequently decompose into the product, the enzyme and the metal ion. So M plus S is give MS, E plus MS is give EMS, EMS give product plus E plus M. Now, third condition, the enzyme react with the substrate to form enzyme substrate complex ES, which then combine with the metal ion to give the enzyme substrate metal complex ESM, and this ESM decompose subsequently to finish the product. So E plus S it give ES, ES plus M it give ESM, ESM undergo dissociation it give product plus E plus M. So, my friend, out of these three mechanisms, the first is followed by most of the metal enzyme complex system. It finds support in the kinetic studies and determination of equilibrium constant for the complex involved. The second mechanism, although less commonly reported, is followed in a number of cases, like inorganic pyrophosphatase, hexokinase, creatinine, phosphokinases. The enzyme is often inactive towards simple derivative of this substrate. In many cases, my friend, the simple replacement of hydrogen atom by a methyl group completely block the reaction, and the elucidation of the chemical mechanism by which enzyme catalyze reaction is a difficult problem. So the most commonly used theory for the providing a detailed mechanism in these cases is play affinity theory of Bergman, which may be some of the enzyme interact with the multiple site on the substrate. So Calvin emphasizes the suitability of the coordination process for the finishing with this second kind of action. The involvement of metal complexes in the enzymatic reaction was pointed out by Hellman, while Smith postulated the formation of mixed complexes as intermediate in metal activated enzymatic reaction. And Smith worked on leucine aminopeptidase and pointed out that enzyme functions through the formation of magnesium 2 plus or magnesium 2 plus activated complex. For example, glycinyl L-leucinamide forms manganese 2 activated enzyme leucine aminopeptidase complex. So, apoenzyme combined with the manganese and forming the complex. Now, orientation in steric effect. Smith listed the factor resulted from the metal chelates involved in determining the specificity of these enzymes. So, the substrate should have a specific polar group, and these must combine with the enzyme during hydrolysis and presumed to form chelate with the metal. The substrate must have the characteristic orientation, preferably the L orientation. The rate of hydrolysis of L-leucinamide is about 1,000 times faster than that of D-leucinamide. So the substituents affect the energy of the peptide bond. Thus, with the increase in the acid strength of the carboxyl group of the acid involved in the peptide link, and there is an increase in the number of hydrolysis. My friend, the nature of the alkyl group attached to the alpha carbon atom of the peptide also affect the rate of hydrolysis. So it is believed that the biological response of enzyme is the result of its interaction with the receptor. Receptor may be considered as a locus on the plasmic membrane with a structure which facilitates its preferential and selective contact with enzyme molecule at the surface or inside the cell affected. Recent studies have shown them proteinous in nature, usually a membrane protein, and they may be regarded as a chemical group on the receptor, photoplasmic micromolecules, which give recognition site for the molecules having a specific structure, the drug molecules. The shape of the enzyme molecule in three-dimensional space is crucial for union with the receptor. My friend, molecules with a fixed conformations may only get fitted in this site, and this suggests that the enzyme and their receptors share complementary structure which help an enzyme to recognize its receptor. So it is said receptor response are genetically determined, and this is somewhat similar to the lock and key arrangement. Enzyme substrate interaction. It is believed that metal ion helps in the giving proper orientation of the enzyme in the receptor site and reducing strain. Now, my friend, mode of enhancement of rate of bond cleavage. There is a group of enzymes which are involved in the natural process of bond formation and bond cleavage, such as during cleavage of peptide bond, the metabolic decomposition of protein into amino acid. The enzyme involved are endopeptidases, and such as chymotrypsin, calcium 2 plus complex, exopeptidase, bivalent metal complexes, dipeptidase such as glycyl glycine dipeptidase, copper 2 plus complex, or glycyl leucine dipeptidase, zinc 2 plus or manganese 2 plus complex, and carboxypeptidase, manganese 2 plus complexes. My friend, during carboxylation and decarboxylation reaction, the addition and removal of carbon dioxide, such as conversion of oxalosuccinic acid to alpha ketoglutaric acid and of alpha ketoglutaric acid to succinic acid. The carboxylase enzyme involved are magnesium and manganese complexes, while carbonic anhydrase is a zinc 2 complexes. During the phosphorylation reaction, synthesis or destruction of the phosphate bond, the energy source of biochemical reactions such as conversion of ATP to ADP by phosphorylase manganese 2 plus 2 complex, or the activity of the contractile protein actomyosin magnesium 2 complexes, or biological activity of insulin zinc 2 complexes, a metal enzyme complex function as a catalyst. Similarly, during the other condensation and the cleavage reaction, you know, such as condensation of acetate in the formation of enzyme A, magnesium 2 plus or calcium 2 complexes with oxaloacetic acid in order to form citric acid, or the formation of enolase. In enolase, you know, magnesium 2 plus complexes for them. And during the dehydration of phosphoglyceric acid to phosphophenol pyruvate depends on the metal enzyme complex catalyst. And during this reaction, many modes are involved which enhance the rate of bond cleavage, like my friend acid-base catalysis, covalent catalysis, strain and molecular distortion. Now, acid-base catalysis. What is acid-base catalysis? The reaction which are catalyzed by the presence of an acid or base in the reaction mixture are called acid-base catalysis reaction. In simple, acid-base catalysis reaction. You know, the specific acid-base catalysis reaction are those reaction the rate of which are changed with the change in ion in the solution, while they remain unaffected in the presence of other acid bases. On the contrary, those reactions whose rate are affected by the presence of all acid and bases in the reaction solution are known as general acid-base catalysis reaction. Rotation of the glucose in a solution is a general acid-base catalysis. Hydrolysis of an organic ester or amide is a specific acid catalysis reaction. Thus, acid catalyzed hydrolysis of an amide may be represented as follows. So, I will show you or I will send you this The functional group of enzyme orient in the active center in such a way that they interact with the substrate effectively. In the substrate protein donor and proton